Yeah, good afternoon, good morning. Right now we'll be talking about the notable biochemistry of bacteria. Notable biochemistry of bacteria. What is biochemistry of bacteria? Biochemistry of bacteria are how the bacteria processes different chemicals to enhance its livelihood, to foster its livelihood, to stay alive. Because being alive is a lot of chemistry and physics of everything working together in a way that makes the bacteria stay alive. Like active transport, passive transport, all those stuffs are actually functional biochemistry reaction, and there are different tests that has been carried out with different importance in order to classify bacteria. One is the gram staining. Gram staining actually is a stain that they use to stain the cell wall of a bacteria to know how thick the cell wall is. Because some bacteria have a very thick cell wall, which this bacteria can actually resist the stain of this that is used in gram staining, the gram stain, and once it can resist this gram stain, it retains its color and all those results that happens after testing the bacteria. That's what gram staining is. Gram staining is used to test the thickness of the cell wall of the bacteria and the kind of the kind of chemical that is on that. Because let me tell you, there are two different types of results that is getting after the carried gram stain. There's the gram positive bacteria and there's the gram negative bacteria. The gram positive bacteria are said to have thick peptidoglycan layer, but it doesn't mean they have a very thin wall. Their wall is still thick too, only that is containing another lipopolysaccharide, and this kind of lipopolysaccharide is dangerous. You understand? This gram negative have more of that type of cell wall, and this cell wall can causes or increases violence because it has the ability to evade lysosomes and also lyse other cells at the same time. So it's like a conjugate lysosome which have the ability to lyse and also defends itself from being lysed. You get what I'm saying? So that is the functional reason why they do gram staining. That is the reason why gram staining is done. So you can go through the internet to look at the procedure of carrying out gram staining in the laboratory, because that is very, very important, but won't be in this course because that is just a procedural note on how to carry out the test. But the functional why you should carry out that test when you are doing some research is that some research may not require you to carry out gram staining. If the research is not actually asking you to identify this bacteria based on the ability to resist that gram, there might be a new way of actually detecting more dangerous microorganisms that is a bit different from gram staining, but is still using the same concept of gram staining. That will be another conjugate stain. You get what I'm saying? Now, there's what they call catalase test. Catalase test is used to see if this bacteria can survive in aerobic condition. Now, there's what they call hydrogen peroxide. Hydrogen peroxide is said to be dangerous or lethal against these cells. You get H2O2, but it also comes as an end result. As end result of metabolism with the air. You must be able to pump that out. For the bacteria, they must be able to pump it out in order to be alive. You understand what I'm saying now? So this catalase test gives the ability to convert that into water again. So it converts H2O2 into water. So catalase is an enzyme. So this enzyme gives the bacteria the ability to survive in aerobic condition. Every bacteria containing catalase, which is an enzyme, can survive aerobic condition. So the catalase test is used to see if this bacteria can be alive in aerobic condition or not. So if the bacteria cannot be alive in aerobic condition, then it is catalase negative because it doesn't have catalase. If it can survive, then it has catalase, and it's done by using hydrogen peroxide to whatever. You should read that online too. I'm telling you the functional reason why you should carry this research if it fits to your own research. The reason why they carry out this test. There's what they call coagulase test too. Coagulase test is actually a very important test too. It tests whether this organism have the ability to coagulate blood. Now, if this bacteria that has the ability to coagulate blood, is available, is the one. If that kind of bacteria enter your body, it's going to cause a very dangerous bacteremia disease. You get what I'm saying? Meaning you are going to have a lot of problem in blood. Your blood will be clotting from the inside. A lot of things will happen. You have to take drugs, and it's a very, very dangerous bacteria you get to the human cell. Thanks to our immune system, which is very strong enough, once it's identified such, it goes there to destroy it, including the blood cell and whatever. But don't forget, blood is life. The more blood cell you live, the shorter your lifespan is. So you should make sure you avoid coagulants kind of organism. So this test is used to test if this bacteria can coagulate blood or not. No, you get what I'm saying now? So once it happens that way, coagulase test you carry out to that. So you must always use coagulase test when you are afraid of bacteria that might or you are trying to study if the bacteria can actually cause hemocoagulation or not. You get what I'm saying? There's the UAS test. UAS test, you know what they call urea? Urea is actually part of what comes out in the urine. You get urea can be negative against this bacteria itself. You get what I'm saying? So if this bacteria has the ability to resist urea, you get what I'm saying now, then it is more violent, because urea is a kind of toxin specifically created that it has a dangerous effect on the body. That is why it is also restricted, but not as dangerous as it is created intentionally by the body to actually use to fight against microorganisms, while once it is getting too much, it gets restricted by the body immediately, because at a level of concentration it becomes harmful to the human body. So once that happens, if this bacteria has the ability to resist this urea, then it has UAs, and then that is what UAs test for. So if it is UAs positive, then it can actually degrade urea, and that's why it is UAs positive. If not, it cannot. Then if it cannot, then you should not be afraid. It's your immune system can still fight it using urea. But if it is urea positive, then your body cannot fight it using urea concentration. That is that about that. There is the endospore stain. Endospore stain is also a very, very important thing. See, there are some bacillus species that has endospore, and with this endospore they can resist adverse condition. Once there is a big attack, you get what I'm saying, it will just turn into a docile phase. You get, once it turns into the docile phase, it will be resistance. The docile phase, you get, or spore phase, is that spore that gives it resistance. So it will create a cyst for itself, hide inside that spore, and become non-vegetative during the period at which it is in that adverse condition. So let's say an endospore former enter your body. Once it enters your body, you take drugs that will attack it, and then it will become an endospore. You get what I'm saying? It's going to form endospore, and it will still be alive. Be thankful and be prayerful enough that, okay, you have excretory system that is trying to sieve out everything that is not good for your body. That is what happens when it becomes that state. Then the urinary system now flushes it out. If it is in the urinary system, if it is in the digestive system, that one now flushes it out through the poo-poo. Yes. So be thankful that you can actually secrete all those stuff out of your body. But if not, the importance is that these bacteria are dangerous. They are dangerous. If you are not good enough, or you don't use the right procedure to flush them out of your body, they will just resist the chemicals and come back alive in your body and still continue to cause the problem they cause. Not because they want to cause it, but because their byproduct and your cellular body are not compatible. That is why. Then there is the heat shock. Now, heat shock is also one test that is used to test if the bacteria can form spore or cyst or whatever. This heat shock is, you are trying to test how much heat this bacteria can resist. You get what I'm saying now? And the kind of heat it can resist. Sometimes you can do advanced heat shock where you put the temperature at a very high degree and see if the bacteria will now grow after you've actually made it subdime and come down again and plated on plate. And also you can do heat shock in a way that you just remove each and refrigerate it at a very, very low temperature too. That's another form of heat shock. You get what I'm saying? Then there's intermittent shocking, where you heat it up, you suddenly cool it down with the refrigerator, ice refrigerator, and then you heat it up again, and then suddenly cool it down again, like you suddenly freeze. So just see which kind of methods that can be used physically to actually go against this bacteria in a real life perspective. Like you just expose the body into cold, and then it's cold shock, and then remove it from cold and warm it up again, just to see if it will fight that bacteria or viruses and disrupt its metabolism. There's what they call nutrient starvation test. Our body deprive organisms of particular nutrients depending on the kind of organism, if it's able to identify it. And this is why we lost our appetite when we fall sick. You understand what I'm saying now? So nutrient starvation is a test you use to see if this bacteria and what the bacteria is going to produce when you starve it of a particular nutrient. You got what I'm saying? So that's what nutrient starvation test is all about. And then there's antibiotic assay, which is used to test whether this chemical drugs is effective against that bacteria or not. All these things are tests on the biochemistry of the organisms, and they are very important. You can go online to search them one after the other to know how to carry them out. But in this course, all I'm telling you is why they are carried out, why you should want or would not want to carry it out, depending on the focus of your own research. There are many more biochemical tests. There's the VP, there's this, there's that, there's different types, but I'm not talking much about them because for the level of bacteriological research and undergraduate level, you understand, you don't really need to know all this because most of your research will be focused within this range. Okay, so I'm just letting you to know the functional application of this range. Once you master using this one, then you can advance to a higher level of research. You get. Thank you very much. God bless you, and me too.